So with this method, we've been able to go on and actually sequence the genome of this Denisovan from the finger bone to over 30x coverage. So we have now a quite complete version of this genome, that is of the 1.8 gigabases of the genome that is possible to map short fragments to, so excluding duplicated regions, etc. But for those parts of the genome, we have the entire sequence. So that allows us to begin to do quite amazing things. For example, we can see that this old individual sort of lack substitutions. Because it's an old individual, we would expect it to not have as much substitutions as people today. And indeed, that is the case. If we compare to present-day humans, they have about 9% less substitutions. So if we now sort of arbitrarily, more or less, say that there was 6.5 million years to the common ancestor with the chimp, this translates to somewhere between 60 and 80,000 years ago for this bone. And this bone is, of course, so small, so you cannot carbon date it. But by the lack of substitutions, we can now start to gauge sort of an approximately age of the bone. This is still very tentative, but I think it's an indication of what will come in the future. We will probably be able to date beyond the range of carbon dating and actually often using less samples than for carbon dating how old things are when we really understand how mutations accumulate in the human genome. You can, of course, also distinguish the two alleles in this genome now, in this about 50,000-year-old individual. So you can look at heterozygosity, how much variation is around, and compare it to present-day people. So we have Africans today, have more variation, as is said, than people in Eurasia. And this guy from Denisova has a lot less variation, about 20% of that seen in Africa or 30% of that seen in Europe. And it's not due to recent interbreeding, it's not that there are huge, huge tracts of homozygosity, but it's probably typical of the population at the time. You can also then reconstruct for each part of the genome the time to a most recent common ancestor between the two alleles. So if you look at these two alleles, you can sort of in a window in the genome reconstruct the time back to when these two alleles have a common ancestor. And you can walk across the genome and do that for many different parts. And you can then look at the distribution of depth here and use an approach that Heng Li and Richard Durbin had developed to translate that into an approximate population size back in time. So you can infer the population size by the fact that where many of these coalesce, it's probably a time when the population was small, so many parts of the genome share a common ancestor at that time. So the population size in this little sketch would be smaller here. So if we do that and look back in time to in present-day people, we have more variation in Africans than in non-Africans. They go back to time when everyone living today, unfortunately the pointer doesn't really work, but you come back to a time when everybody had the same population size, and you see a time of expansion of the population there. That's probably the expansion of modern human before one leaves Africa and Africans start differentiating from non-Africans. The red line there is then the Denisovan. And you will see that it has a very different population history. When modern humans expand, this population actually decreases in size until then, some time ago, when this individual lived. So it will be very interesting in the future when we have a Neanderthal high-coverage genome to follow the population history of Neanderthals in the same way, and we actually have a Neanderthal genome almost finished by now to the same quality as this. So just to give you a feeling for that, with this high coverage, you can begin to look at many things that may be interesting. One interesting thing is, for example, that the human chromosome 2 is a fusion of two smaller chromosomes that all the apes have. That fusion, of course, resulted in the fusion of two telomeres here. So the telomere repeats are then head to tail at that position. It's the only place in our genome where we have these telomeres looking in this way to each other. So we can look among our reads, and indeed we find in the Denisovan these head-to-tail fragments that we don't see if we look in the apes, for example. So this shows quite clearly that this fusion of chromosome 2 is something that the Denisovan already shared with us. It was sort of a tempting speculation that if that fusion is more recent, that could have been responsible for reproductive isolation between archaic humans and present-day humans. We can ask, just as for the Neanderthals, have the Denisovans contributed to present-day people? We've done that by comparing to 48 populations collected from around the world to study human diversity. And we can separate then these populations essentially in two axes. On the y-axis according to Neanderthal contribution, and on the x-axis according to Denisovan contribution. So we separate then Africans from non-Africans in terms of this Neanderthal contribution, but we find two of these 48 populations stand out. One is Papua New Guinea, and another is Bougainville, another island in the Pacific, that have a special relationship with the Denisovan, it seems. So this was very surprising. We have this Siberian individual, and they seem to have contributed to people out in the Pacific. So we've studied that more carefully in many other populations, and we actually find no contribution in mainland Asia, but find it specifically out here in Melanesia, in Aboriginal Australians, Fiji, and so on. So the model that this results in is then that we believe modern humans arrived in Southeast Asia, carrying with them 2 or 3% of Neanderthal DNA in their genome. Somewhere there, in Southeast Asia, they run into Denisovans and mix with them, actually to the extent that they get about twice as much from the Denisovans as when they got them from Neanderthals, about 5%, and carry that with them out into the Pacific. So I think it's sort of fascinating and also frustrating in a way that from a little tiny fragment of bone, such as this, you can reconstruct this much of the history of a population, but we have no other remains. We have no clue how this individual looked, what their morphology was like. It's sort of an indication what will be in the future in paleontology, I think, that you from tiny samples can learn a lot by studying their genomes. So if we, for a minute then, think about the model of recent human evolution that come from studying these archaic genomes, it's then that Denisovans and Neanderthals have a common origin somewhere in the order of 500,000 years ago or so, probably in Africa. They come out of Africa. In the Western Eurasia, they evolved to what we call Neanderthals. And in the East, they evolved to what we call Denisovans. That's not to say that there were only Denisovans in the East. There were clearly other forms too, maybe many other forms. We also don't know where the border between Denisovans and Neanderthals were. It probably varied over time. But we know that in this cave here in Siberia, in Denisova cave, at some times there were Denisovans there, at some other times there were Neanderthals there, because we now also have a Neanderthal bone from that cave. And then somewhere 100,000 years ago, modern humans appear somewhere in Africa. Around 50,000 years, they seriously start spreading out of Africa. Probably in the Middle East, they then meet Neanderthals and mix with them and continue to spread across Eurasia. Perhaps there is another event of mixture somewhere in the East here. In Southeast Asia, then run into Denisovans or close relatives of Denisovans, mix with them, and move on out into the Pacific. These archaic forms of humans then become extinct and disappear, but they live on then a little bit today in many of us, in that 2.5% of people outside Africa then have 2.5% of the genomes of people outside Africa come from Neanderthals, and in the Pacific something like 7 or 8%. I think that we now studied 2 archaic genomes, found 2 cases of mixing. Probably we will find more when one finds other groups, particularly perhaps in China. I think one will probably find other cases of admixture. I think that inside Africa, modern humans, of course, also spread around, and it's very reasonable to assume that they mix there also with earlier forms of humans. So looking at this scheme again then, it's very clear that what we sort of falsified is the absolute total replacement. There has been mixing with these earlier forms, but it hasn't been that much. Actually, it has been something like 2.5% of the genome all over Eurasia, as you said, something another 5% in Melanesia. So if we should sort of dub a name for this model of modern human origins, I like to sort of call it leaky replacement. The big picture is replacement, but there is some gene flow from these earlier forms into present-day humans. So in a way, this has then been talking about the power of genomics in this little field, human evolution, what we can do by studying genomes, leveraging the Human Genome Project and what we know about variation in present-day humans, and adding in genomes from extinct forms of humans. We were also sort of encouraged to talk about the promise for the future. So I have to promise something. And I think what really fascinates me is actually thinking now about the few changes that we do have in present-day humans where we do differ from Neanderthals and Denisovans. So thinking about changes that happened here, that we all share today, but where the Neanderthals look like the chimpanzees and the other apes. So really new things in humans that spread to be present in everyone today, no matter where we live. So those changes that happened here in the last, say, 200, 300, 400,000 years of human history. Why would those changes be particularly interesting? Well, I think they would be particularly interesting because there were some things that really changed with fully modern humans. The Neanderthals, for example, and Denisovans were very closely related to us, as we saw. Yet they lived for 400,000 years without ever sort of developing technology that started changing rapidly. They made pretty much the same stone tools 400,000 years ago and 30,000 years ago when they became extinct. Whereas with modern humans, in less than 100,000 years, technology started changing very, very rapidly, became regionalized in the world, and we all know how it has taken off. We can also discuss for a long time what art is, what's the essence of art, but at least art that we, as present-day humans, immediately recognize as art comes only with fully modern humans. And of course, the colonization of the entire globe, spreading to every habitable part of the Earth, comes only in the last 50,000 years of human history after these other forms have disappeared. So there's truly something different with the behavior of fully modern humans. So I think what is really fascinating is a catalog of new features in the human genome where we differ, all of us differ from these other groups. And since a few months we have such a catalog now and can start looking at it when we compare it to the Denisovan high-coverage genome. The fascinating thing is that that's not a very long list to look at. It's in the order of 100,000 changes that, to the best of our knowledge, are fixed among all humans today. It increases to about 200,000 changes if we say 95% of people today carry them. So that translates to not very many splice sites and regulatory sequences and so on that have changed, and only 260 amino acid changes in proteins. But now we run into an area where I think we have an appalling lack of power in genomics today. And that is our ability to, from such a change, now predict its phenotypic consequences. So we can just start to try to approach that by leveraging the things that Eric talked so much about in his talk. We can, for example, look at which of those 260 amino acid changes affect genes where we know about Mendelian diseases, other mutations in these genes, and what effects they have. So we have then 34 such genes that are involved in Mendelian disease that have such fixed changes in them. And it might be interesting to note that 4 of these 34 genes affect diseases of the skin. It's perhaps not so surprising that something in the morphology or physiology of the skin has changed recently in human history. What was very surprising to me was that actually 6 of these genes are involved in diseases of the eye. So it may be that something in human vision has changed relative to our archaic relatives although no one had expected that, I think. Another way to prioritize these are looking at conservation, as Eric also described. So we can look at positions in proteins that are conserved among all monkeys, all apes, and in Neanderthals and Denisovans, yet changed in modern humans, and changed so that it's swept through to encompass everyone, all 7-8 billion people that live on the planet today. So there are then 23 genes that carry such changes at positions and individual amino acids that are extremely conserved, yet changed in modern humans and swept through. So if we do that, we have a list of just 23 proteins. 2 of them are involved in axonal and dendritic growth, so how nerve cells grow out connections to other nerve cells. 2 of them are involved in synaptic transmission, so how nerve cells communicate with each other, and 2 of them affect genes that have risk alleles for autism in them. So all these things are things that have in some form to do with connections between nerve cells. So it's possible, as a hypothesis, that something in sort of cognitive development or sociality has changed here. But this is, of course, something one will have to study in the future in much, much more detail. But I think then that the systematic analysis of this catalog of things will be extremely interesting. So sort of to then find out what is really different between modern human and our closest extinct relatives, what made the human condition, the modern human condition, possible. So a promise among many, many promises from genomics would then be to that we might understand some of the genomic basis for the modern human condition in the future. And with that, I should just mention that there are many, many people involved in this, a lot of present and past collaborators in Leipzig who produce these genomes and develop the technologies that makes it possible. I can't mention individuals as for what are presented in a talk. Matthias Meyer, with this new library technology, has been very instrumental. For the analysis of the genomes, there are many groups around the world involved in the consortium to do this. I should mention particularly Janet Kels, who coordinates all the bioinformatics in Leipzig and provides it to the consortium, and particularly then for the population genetic analysis, Monte Slatkin and his group in Berkeley, and David Reich and his group at the Broad. And I thank you for your attention.
Thanks so much, Svante, for that fantastic talk. And we have a few minutes for questions. Come to the microphone. Yeah.
Yes. Hi. Thank you very much. I really enjoyed that. Could you give us a sense of the populations we were actually talking about in the Neanderthals and the others? We think of 5 or 6 billion as the norm, but it wasn't 100 years ago. And what was it like 500,000 years ago for each of those populations where this mixing and was occurring?
Yes, so population sizes, I think paleontologists have a very big problem in inferring population size from the number of sites and bones they find. And I'm not really an expert in that. But if we say that effective population size, something that geneticists tend to think about, which is also a very theoretical concept, right, of essentially the population size that over a long time, and if it was panmictic, would give rise to the variation we see now in the population. That number for modern humans is in order of 10,000. For the Denisovans, where we can look at it, it's about 20% of that, so smaller than modern humans. And for Neanderthals, we have some estimate of it from sort of lower coverage sequencing from 4 different individuals now, and it's in the same order. So I think whatever modern humans sort of long-term size is, these guys have had even less size, probably because they've gone through bottlenecks in at least 2 glaciations during their history. But yes, I would think we're talking about not over 1 million individuals at any one time, for sure. But that's totally from the top of my head.
So when you look at the unique aspects of humans versus these other human as well in evolution, you emphasize coding sequences, but I would anticipate that the regulatory sequences is where you would see the biggest difference. Is it just you don't have enough information to look at that, or is that incorrect? The regulatory sequence changes are not the major contributor.
I think regulatory sequences may very well be very important. So we have in that list there 36 sort of well-defined ENCODE transcription factor binding sites where we have a change that seems to change it and it's fixed in humans. It may well be that those 36 ones are the ones that will be really interesting in the end. It's just so tempting to present the protein coding one since we know a bit more about what systems might be involved and so on.
Yes, Pavel Hamid, Montreal. Interestingly, we have shown as an example of the early art the obesity phenotype. No, this is actually important because I think that obesity, hypertension, diabetes are not only features of the modern humans, but that were present for a long time. Actually, all the examples of the early art are pictures of obese women. And can that approach, what you are showing, help us in the diseases when the genes and environment are playing together? How this moving outside of Africa, because some features are still same. Some of the diseases, hypertension prevalence is even higher in Africa. How that's modified with the change in environment and diversity of the genome?
Yes, I think it's of course a situation that those diseases we call complex, where many genes are involved, also have a huge, huge environmental factor. So things such as obesity is clearly, of course, something where environment play a big role. I think, yes, it will be interesting to look at haplotypes associated with disease, if some of them come from these archaic groups, or some are these that have swept through and may have had some advantage, but in the present day environment sort of lead to what we define as diseases. I think it will be in that way it goes. And the fact that these people look obese is probably perhaps more an aesthetic expression from the time of an ideal, I don't know.
Thank you.
So let's please join me in thanking Svante.